So bee vectoring technology. So he's claiming that you can get bees to get like coated, their body coated with a patented mixture of microbes that can vary depending on your crop, and that they will serve as a vector to get your biocontrols, essentially, your biocontrol agents from plant to plant. So Vectorite is an organic carrier agent which bonds with stacked biocontrols. So they have like a base medium, a carrier agent, and then you mix in different cocktails of microbes, depending on what crop is being targeted. The bees fly to the crops and deliver the beneficial microbe in an efficient way. Microbe colonizes the plant, thus protecting the crop against various pests and diseases. So obviously, we're thinking about different flowering plants, and that the bees, of course, they're going to the flowers in order to, in the process of getting nectar or pollen, they're pollinating the different plants. And so they're claiming that through this process, you can deliver microbial biocontrol agents. And their argument is, since it's the flower that you need to protect because that has to turn into the fruit, which we then harvest and eat, it makes sense to have the bees involved so that, as they're crawling over the flower and maybe even the nearby leaves, they're depositing the biocontrol agent. The first thing that kind of popped into my head is dose, like how much dose can really be applied. But I suppose, here they have like a beehives here, and then I think they have like the vectorite carrier agent in the beehive somewhere where they're confident the bees are going to get well coated. Um, so I think there's something to this. It's worth exploring. And then I started looking for has there been any science on this? And this is one that I found. This was published in 2019. And it caught my eye, of course, because I work with Streptomyces bacteria. So I was very interested in this idea. But this is essentially, again, the same idea of using pollinating bees as vectors. The Streptomyces are biocontrol agents in this case, and they were working with strawberry plants. So I wanted to just go over a little bit of this with you. Um, but this was an endophytic Streptomyces bacteria. So it was known to be in the strawberry plant. And they found that it could colonize the rhizosphere. It could hang out on the flowers. It can get transferred by the bees. And not only that, it was protecting the bees themselves from their pathogens. So it was this interesting added positive impact. So here, what they were doing, they worked with the strawberry plants, the Streptomyces bacteria, and they isolated them from flowers and pollen initially and started working with them. And they discovered that they can produce antibiotics that can inhibit Botrytis cinerea, which is a fungus that causes a variety of plant diseases. And certainly strawberries are easily colonized, unfortunately, with botrytis. So this plate on the left is the fungus growing across the plate. And then on the right, those streak lines that you see, those are two different Streptomyces strains. They usually hear their letters and numbers. And then the botrytis fungus, you can see, is greatly inhibited. So these are agar plate assays that are commonly done to test inhibitory profiles and to discover biocontrol agents. So they were confident that the Streptomyces that they isolated was, at least in agar plate assays, it definitely could inhibit. And then they, of course, wanted to add in the bee pollinators. And then they also recognized that the bees themselves have pathogens that make the bees sick. So they were also curious to see if maybe the Streptomyces bacteria could be helping out the bees directly by helping them fight off the pathogen of the bees. So they had some data where they showed, they just said actinobacteria, which is a group that the Streptomyces belong to. So the actinobacteria in their study, almost half of the total bacteria of the flowers was the Streptomyces. So that's a pretty high percentage. That's pretty unusual. And then in the pollen too, that, 43%, that's also really high. So that is definitely attractive that the Streptomyces, or at least the actinobacteria group that the Streptomyces belong to, so much of the total bacteria in the flowers and pollen, that's a really positive sign to keep working with these bacteria and see what they can do. So here's some of their results where they were following time on the x-axis here. And then there's a couple of things being shown on the y-axis. You have the Streptomyces, OTU is operational taxonomic units. Basically, it's their abundance, if you want to think of it that way. And then on the other y-axis, you have the disease incidence, so the amount of botrytis, the incidence of that. And you can see that so the black here, you can see this is the Streptomyces, high abundance, and then you can see that they went down over at around 8 to 10 weeks, their numbers went down. And the square one is the other Streptomyces, and you can see they came up a little later, but also went down around 14. And then you can see this really interesting disease incidence. You see when these two Streptomyces were at high levels, you see the circles here, the clear circles, very, very low disease incidence. And then after these two, their population went down, all of a sudden you see this big spike in disease incidence. So it's very compelling data to show that the bacteria's presence at high enough population levels was keeping the disease in check. But then when their levels started to go down, it allowed the pathogen to get the upper hand. So high density to low density numbers, and once they reached low density, the pathogen was able to gain a foothold and cause some issues. So that was, again, of course you want to ask, so, here they could show that their numbers were, crashing after 10, 12 weeks. Can they, like how long can they persist? So they were doing studies like this where they could show that two different biocontrol, the Streptomyces strains, were at really high numbers actually. And they persisted. You could see from the day of inoculation at day 0. And then after 5 days, there was not much decline. And again, you want your biocontrol agents to persist. So these are some of the studies where they're just trying to see, if we use these bacteria, are they going to be able to hang in there or not? And then they had to show whether or not the Streptomyces could actually get transferred to the bees. And so they would have these bowls in the hive with, in this case, they chose that particular Streptomyces strain. And they could show that, so this is the concentration of the two different bacteria in the stock. So that's kind of like your control. That's what they started with in the bowl. And then you can see that the honey bee body definitely had some good colonization and even into the gut. So they know that the honeybee was able to swallow some of that, and which if you want the honeybee to be protected, probably some internal ingestion is a good idea. And the honeybees survived and everything was fine. And so you can see that the populations did drop a little bit compared to the stock culture, but still you have decent amounts on the bee and in the bee. So they were wondering, like, okay, when we initially isolated these Streptomyces, we got them from the flower and pollen when, we were just asking the question, what kind of microbes are in flowers and pollen? But then they were like, okay, we know it's an endophytic streptomyces as well. They can be inside the plant. So then they investigated like how's the plant getting exposed to the streptomyces and all of that. So they did some really nice studies, tagging them with dyes and following, what happens if you put these streptomyces in the soil. Are they going to get into the root? And sure enough, that's what they're showing. You can see the root. Well, it's kind of hard with the lighting in here. Can you see the root? Okay, so the red is all the streptomyces in the root. And this is with one of the streptomyces on the left here, and then this is the control where they didn't inoculate the soil, and you can see an absence of the red. So the red is the bacteria, and so you're seeing that it's moving from, it got into the root, and then they were able to identify that it was in the stem as well. So they were just trying to gather evidence to show that if you were to inoculate these biocontrol agents into the soil, they should be able to go into the plant endophytic and ultimately end up at the flower and in the pollen. So then they had to ask, okay, so when bees come to the flowers, if there's streptomyces there, can they successfully transfer them to another flower of the same species? And so that's what they were trying in this experiment, where they had a control group, which was untreated, and then treated flowers, and showing whether or not the bees were capable of transferring. And you can see that it did show up, the bacteria did show up on the body. Oh, and they blocked, did they show that? Oh yeah, so they capped one of the, just to show that, I don't know, that seems kind of a weird control, but anyways, you can show that they were getting transferred to the bee's body. And then on the far right, those figure C, that is the test on whether or not those streptomyces could help the bees against their own pathogens. And you can see that if you just have the bee and you inoculate the pathogen, the bees have a high mortality rate. But then here you can see all three, the bee, the biocontrol agent, and the pathogen, a significant drop in mortality. And then they're also just trying to show that the streptomyces aren't causing any issues with, they're not themselves making the bee sick in any way. So then they're saying, okay, well, what is it going to be good for the strawberry plants? They did this experiment in the greenhouse, and they were looking here at abundance of a variety of different phyla of bacteria. So the legend is over here. And the black area is the actinobacteria, where that's the group the streptomyces belong into. So you can see that they're there, but they're in this experiment, they didn't show up at super high levels. The yellow, definitely not the proteobacteria, those are showing up at really high levels. But when on this side, they were showing, yep, we have the actinobacteria showing up, they're there, and then they were tracking disease of the strawberries in the greenhouse. And you can see that compared to over here, on this one, there were no actinobacteria in that sampling. Okay, so there's disappearance of those. They didn't have those in this growth cycle. And so the absence of the actinobacteria in this other set of experiments, you can see the disease incidence went up. And so, by 10 weeks, the disease incidence was up to about 35%. Whereas here with the actinobacteria, we got 12%, and that kind of leveled off. So they're saying that, it's showing that the presence of the actinobacteria does seem to be impacting disease incidents on strawberry plant. And then here they were taking air samples in the greenhouse. Just to demonstrate that botrytis really was there. It was there. It wasn't like they were in a pristine greenhouse. And so they were just demonstrating that, yes, there is botrytis there. And the presence of the actinobacteria does seem to keep the levels down. So they were able to show, they did some really nice studies here in the greenhouse that, yep, it can get from the soil into the roots, into the be an endophyte, good all the way up to the flower and pollen, and it can impact and reduce disease against botrytis. You can get plant to plant transfer. And even the honeybee was getting helped by having the streptomyces on and in it against its own pathogens. So they're saying, yeah, the plant served as a host. You have partners in this whole thing. You have the insect and the bacteria and the plant, and even the insect pathogens are getting the bees benefiting from that. So really nice study showing that you have this really interesting interaction where it's kind of a win-win-win all the way around. And so this would, again, coming back to the idea of the bee vectoring technology, they might be onto something. I don't know. Especially when you think about a lot of the greenhouse crops. It's really hard to keep botrytis out once it gets in. That it lurks in the greenhouse. So having a way to um, I don't know, to do this. And I think the next study they were going to do is out into the, field study to see how it would impact. But my only concern is, bees are small. There's only so much bacteria that can fit on those little bodies. So you have to have some pretty potent biocontrol agents. But I mean, they were able to show with that study, they could show that there was nice decrease. So you might hear more about this. It sounds like it's a pretty new thing. Has anyone heard about this before? Bee vectoring technology? So I think it's going to be something maybe coming online as another method, not, we're going to have to use a lot of different methods in this whole idea of how do we grow enough food for everybody. So, all right. One last concept to think about. This term is becoming big catchphrase, precision agriculture or precision farming, where they're saying we need to be more precise on how we use things instead of just massive dosing of fields of, they're saying, why don't we use drones and AI and fancy sensors, robotics in order to grow food a little bit more sustainably for sure, and just in a smarter way where we can get a better return on investment. And so all of these can be used for doing soil and field analysis. Of course, drones can get aerial views of things. They're starting to use drones to look at the health of fields of plants, where you can determine if there are patches of the acreage that are in trouble, and you'll be able to more quickly, hopefully, help the plants. They can be used for actually planting seeds as well, irrigation and crop spraying, investigating plant health status. They're designing all of these sensors and drones in order to do this. And they're really getting into this idea of precision irrigation, where you don't just spray water from these huge above ground sprinklers. You do more drip control or even just point application control of irrigation if needed, depending on the plant. Same with pest eradication. They're looking at what would be the word? uh, basically, would they be using like a flamethrower type thing? I guess that's what I'm thinking of, a tiny flamethrower. I've seen video of that like hitting weeds where the rest of the crop is left alone.
Well, I saw some guy on Instagram make like a mosquito shooter, like a fly shooter where it would shoot the flies before it got to the food. And it's pretty weird.
What would it shoot the fly with?
It was a laser,
A laser? That's laser technology. That's what I was trying to think about.
a little like anti-air laser.
Yeah.
And
And they're doing that with like weeds that show up in the rows. They'll hit it with a laser light and really interesting video of the plant blowing up and turning into fire. So they're trying these out. I don't know how much is in current production. I was looking for some stats on that and finding a hard time with some of this. I know that they're using drones a lot, but some of the more fancy machines, I don't know. Oh, robotic harvest pickers. That's another thing that they're looking at. I'll show you a picture of that in a minute. Uh, Tractors that don't need a driver. They're really interested in that idea, and that is hitting the market. I have seen some of those being used. Um, and then just the idea of, like I said before, we hear a lot about sustainability, and the term regenerative is like almost a step above it, where they're saying we don't want to keep things status quo. We want to actually make things better. So make the soil better than it was before we planted, for example, make the air even more better quality. And so vertical farming is catching on globally. This is something that there was a student in the indoor egg program that was from over in Turkey. And he was so excited to get back to his country so that he could start up a business on vertical farming because he said they were actually like wanting him to come back and start up that business. Plant breeding will continue. There have been plant breeding programs for decades and decades, but that won't go away. That's still a viable way of trying to get plants well suited for their different habitats. And of course, now we have better technology with CRISPR and just some of the genetic editing that we have. The technology is better. So that can play a role as well, in addition to selective breeding. And then the other big idea is don't be so secretive about the data. Why can't we share that so that it's just not one company that has all the data and that's it. They're saying we need open access to, all the data on growing plants in different habitats under different conditions, different soils. We need that and we need a lot of it so that we can run really strong statistics on it. And that was the other thing too, when I was at the last plant conference, so many of the businesses that were giving presentations were like desperate for people that could do bioinformatics and biostatistics and people who can do, are really comfortable with computers, but also really good at statistical analysis. They were like so desperate they kept saying, make sure your students get some biostats training before they graduate, because that is big data. It's just going to get bigger and bigger and hopefully more open access. So and then they were also at that conference too, they were also saying how important it is for people from different disciplines to work together collaboratively, interdisciplinary, and that it just a lot more sharing of ideas, working together. They said that's really how we're going to solve this. No one group of scientists can do this. We need effort by everybody. And so there's a lot of different approaches that they're coming at this problem with, which is great. So there's the robotic harvest picker up top. That's a drone. And I don't know if you can see, but it actually has this arm and then like a kind of a fancy suction cup type thing where it grabs I think those are apples. And I saw a video of this where it would grab the apple and then fly over to the truck that had the, the bed for where they could deposit the apples. And so they had like a suite of these drones that were hovering above the truck, and they were picking and dumping, um, and they were interviewing the person who owned that farm, and he said that there were actually several different drones that can do this type of thing. And so there are competitors. It's not just one company, but he liked this one in particular because it was not damaging to the produce, and pretty efficient in terms of how quickly it could do it. And then that's an example of a driverless tractor, which they're making good headway on. And then, of course, drone technology. And then, of course, we've already talked, they're getting into this idea of using sound when you're growing crops, to benefit the plant growth and yield. And the VOCs, they're looking at that, chemicals like the hormones, priming them to start defending themselves ahead of time against any pathogens that might be in there. So they're also devising different sensors to try to pick up on plant diseases quicker, so that you don't have to wait for a pretty big infestation before you go, oh, it looks like something's going on here. And so the idea of priming plants right as soon as you start to see that there's any pathogens lurking could be a big step. And then, of course, all the cool research that's going on right now with how to work with LED lights in particular, and different lighting manipulations, so that you can get the crops that much better at growth and health and nutrient density. Have you heard about this one? So this is pretty amazing. Svalbard Global Seed Bank up in the Arctic Circle. And this is in Norway. Technically, it's Norway, the country. And they have, inside this building is 13 years of agricultural history. So it's basically seeds from all over the world's largest collection of agricultural biodiversity. So it's different seeds. Its nickname is the Doomsday Vault, so that if we have something drastic happening, that we can go back to those seeds and use them. So there's a lot of genetic history in there. Um, really interesting. It's deep into the mountain, and it's so cold that they don't really have to do much because it's so cold up there, and into the permafrost area. And so they have seeds from all over the world stored in there. The only problem is I just saw, I can't remember which publication that was, Newsweek maybe. They're worried because of global climate change that this area might warm up too much and the seeds could be at risk. So far, they said there is warming temperatures there, warmer than it's ever been, but so far, because it's buried that deep into the mountain, so far it's staying, the temperatures are staying cold enough and steady enough, but I don't know. They're worried. They're worried about it. So hopefully it will be okay. Okay. So hopefully there won't be any kind of issue where we have to go and use that out of necessity for massive amounts, but it is nice to know that there are all those seeds there. Okay. So in EduCat, where did it go? I wanted to remind you that there's some readings. Well, where is it? Oh, so there's two videos here. The Sweet Future of Vertical Farming. That's a TED Talk, so please watch that. And then the Futuristic Farms that will feed the world. This is a group in Netherlands. Netherlands made an impact really early on. They have been pushing sustainable growing well before anybody else has, I think. And so this one is they're interviewing this is an indoor grow operation, and it just shows how well they can do it. And they're kind of pioneers. They're amazing. They've really committed to this as a country. They've really committed to working on this idea of sustainable ag. So make sure to watch that one too. All right. Okay. That's all I'm going to say about the future of ag, but why don't we take a break and then, or whatever you want to do, but the next part of the class is for you to work on your second presentation. And then the last topic I have to talk to you about is phytoremediation, which will start probably on Wednesday. Okay?